Yeah, like that's like the wait, are we so it's like so stuff that I like we're not like that.
More of a key. That's
What's your favorite?
definitely help.
No. I actually hate them.
Right.
I know. I would like to say like oh no, I didn't. I did not hear you.
They can help.
I no, I see it like Charlie Brown Thanksgiving.
Okay, so of course you see a brief reminder of where we left off last time. So I'm going to give you a couple minutes to check through your notes, check your understanding. And go ahead and draw me a little graph of what would happen during heat exhaustion. During heat exhaustion, this is issue with the body's thermal regulation. So we end up using a lot of extreme measures to regulate the temperature. So you're going to just draw how temperature control should work. So you're going to draw a negative feedback control system for our regulated variable, set point, and show the error signal. This is a general note. Every now and then I'll have these little review activities on the board as you're filing in. That doesn't mean you're late. You're here by 11:45. You're on time. It's just so we get a little bit of activity to get our juices flowing before settling.
Approximately so in theory, it is and we use all the price. Like, I don't know, but he put on shopping space. No, I like he was just all by himself. What?
Yes, he might have one.
I don't think he has an expand, and she's so bored because he's so. It was kind of strong.
Okay, so it's pretty lively in here. So I'm gonna assume that means we're ready and we're ready to go. Let's walk through where we left off. Okay. So we're looking for this temperature control system. We're gonna try and find our regulated variable. That we want to identify our set point. And we want to identify our error signal. It's going to help us see that this is negative. That's the type of drawing we're going to talk about. We're going to start with some axes. Y and X. Okay. When we're talking about homeostasis, what do we know is on the x-axis? Okay, cool. Heard some whispers. Awesome. So time is over here on our x-axis. So we're tracing something over time. What we're tracing is our y-axis. So this is our regulated variable. Regulated variable goes on the y. And specifically, since we're drawing a specific system, we know that our y-axis is temperature. So all of our measurements on the y-axis are measurements of temperature. So temperature is our regulated variable. Okay. That's how we define that. Okay. Our set point is the value that we're trying to return to. Right, so our value that we're trying to return to is the set point. So that's our 37 degrees Celsius. Okay. Is our set point for temperature. So what we see happening when we're thinking about the regulation here. I'll start drawing, so with blue. Okay. So if we're talking about heat exhaustion, you'd see heat beginning to rise. Okay. So when we have a difference from our set point, that's what we call our error signal. This is our error signal. It's just that difference. Okay. So that's the error signal. Just write ES. Okay. And if all of our regulatory systems are working as they should, we're going to pull that temperature back down. Okay. So we see the error signal decrease until there is no error signal. Okay. And that's the homeostasis piece. And that's the negative feedback, right? We went in one direction, we went up, and then we brought it back down. Those are opposing. So that is how we know this is negative feedback. I'm going to go ahead and draw the opposite direction as well, just to emphasize that point. So let's say we overshoot. We bring our body temperature way too low and eventually want to bring it back up to normal. Right? We would also be producing an error signal here. And so best to think of these kind of in absolute values when you're thinking about error signals and negative feedback. So we still have an error signal. And eventually, as we're pulling back up, getting back towards the set point, it would eventually get smaller until there isn't one. Right, so we had a change in one direction. So we have a change in one direction, going lower, change in the opposite direction back to the set point. So this is still negative feedback. Has to do with the direction of those changes being opposite to each other rather than the numbers that we're talking about. And the set point was that 37. Questions on this piece from last time? That slide. So here's the slide we left off. So we have structures that let us do this regulation. We kind of blew through it at the end, so I want to make sure that we think about this a little more deeply now. Okay, so one thing that you regulate in this image is blood pressure. Blood pressure is a regulated variable. You want it to be that nice 120/80. So a thing that can happen is when you're lying down, if you suddenly go to standing up, your blood pressure does actually fall as all the blood gets pulled by gravity to you. But you have regulatory mechanisms that should, as a reflex, immediately correct this. But if you've ever felt like super woozy when you stand up, this is potentially part of it. You feeling that it takes a little time for that regulation to take place. So when you stand up, your blood pressure falls. Okay. So that first thing that we know, we would call the stimulus. And so a stimulus is always just like a thing that happens to something. Okay, so the stimulus here, the blood pressure is falling. That is the stimulus for our body to respond. In order for our body to respond, we need to sense that stimulus, sense that change. So we have sensors in general, and in this specific case, we would have blood pressure receptors that sense that there's been that change, that there's been that stimulus, that the blood pressure fell. So we have sensors located in our body. So especially like in the arch of the aorta, we have a specific structure that tells you what is the blood pressure, and then also in your carotids. Okay, but more generally, in a general homeostatic mechanism, we have a sensor of some type. So that sensor is somewhere out in the body. Often we're going to want to get this information back to the brain. Okay. So we're going to need nerves to do that. So here we see the sensory nerve fibers drawing that information to our integrating center. In this case, the medulla oblongata, which is a structure in the brainstem. So in the brainstem here, in the brain or the integrating center more generally, we start to plan out a response. We're adding together signals from different parts of the body. We're planning how we're going to fix this. So in order to have an actual change, to reverse our error signal, to do that negative feedback, we're going to need nerves going out from that integrating center. In this case, we're going to go to the heart. Okay, so our heart here is our effector or our effector organ. Okay, and one of the ways that we can improve our response to falling blood pressure is by increasing the heart rate, pumping that blood faster, harder. Okay. And that would return our blood pressure back to normal. So we'd see a rise in blood pressure. So we had two opposite directions, right? The fall, we had our blood pressure going down. This mechanism pulls blood pressure back up, a rise. So this is our negative feedback response. So our general structures would be our various receptors, integrating centers, effector organs, and all our sort of signaling pathways are all going to be involved in maintaining homeostasis. This is just a summary slide of some examples, essentially. So our receptors, we were looking at a blood pressure example in that previous slide. So a blood pressure receptor would be a baroreceptor, like a barometer pressure. We also have thermoreceptors for temperature and chemoreceptors for chemical concentrations of things. Our integrating centers are usually in the brain. They're orchestrating the response. So we may need multiple different effector organs to appropriately adjust to a change, which is why we would need to kind of combine everything together. Our signals, we're really thinking about nerves here. We're allowing our different components of this system to communicate. And so our input signal is coming from that receptor out in the body to the integrating center, often in the brain. Output signal going from that integrating center, possibly in the brain, out to an effector. Previous example, the heart. Chemical messages can also be a pathway for this signaling, which is why I didn't just say nerves. So things like hormones could also be one of our signaling mechanisms here. So when we talk about the endocrine system, we'll see a lot of hormones involved in these signaling pathways that help us mediate our responses, help us respond to changes. Now, our effectors are out in the body and they're responsible for like the direct change that we're going to make. So muscles, and we'll see that we actually have 3 types of muscle we're going to think about. We have smooth muscle, which is located in things like blood vessels. We have striated muscles. Those are the typical muscles you think about, like in your arms and legs. Cardiac muscle and the heart. And we'll also think sometimes about glands. And again, that will come up a lot when we talk about the endocrine system. So we have several more basically diagrams of this same process. This is a significant amount of essentially repetition just to kind of hammer this home if the previous diagrams don't make sense to you. So here we see again, just a generalized sensor, an integrating center, sending signal out to an effector. And we can see that mapped on essentially to that little graph that we drew at the beginning, the time versus our regulated variable. So we can see that as we start to deviate from our set point or our normal range, we're activating that sensor, starting to sense that change. We keep changing potentially until our effector gets activated over here on the bottom side on 2, until we get back to the set point. So this is all just about changing back to the set point once we see a change outside our basically acceptable range for our regulated variable. Our definition of our negative feedback loop then is that we're moving in the opposite direction from the change that we see. Okay, so the change from the set point, we have a word for. Change from the set point is getting smaller. The thing that's getting smaller is our error signal. That was our error signal from the previous set of slides. ES. Okay. So the error signal, that change from the set point is getting smaller, reversing our change. This is a continuous process. So we do see things often in this sort of dynamic fluctuation. So you see like little rises and little falls that stay within a relatively normal range. We're always making these kind of fine adjustments because you're always out doing stuff, right? The air around you isn't always the same temperature. Sometimes you're eating, sometimes you're not. We're always doing things that affect what our set point would be if we didn't make changes using our physiological methods. So here is that body temperature example written out in a slightly more formal form. So we have a normal range where we kind of fluctuate up and down around a set point. So we're always kind of overshooting, getting back to the set point, maybe going a little bit under, but always trying to come around that expected set point. If you take your temperature when you go home today, it might not be exactly 98.6, might not be exactly 37 degrees Celsius. I'm going to be in some normal range, hopefully, unless you're not feeling well. And we might also think about the ways in which we change that temperature. So 2 changes that effector organs can make to change your body temperature that are observable in your life are shivering versus sweating. Okay? probably all know this, that if you get cold a lot around here, right, you start to shiver. So when you're shivering, that vibration essentially from the muscles, you're expending energy to lightly move your muscles to create that movement. And this produces heat as a byproduct. So when you shiver, creating that heat pulls you back up to your set point. And if you overshot your set point, you might start to sweat. Or say you're shivering and then that's pulling your heat back up and you also go into a warmer room. So maybe you're sweating now. So the sweating is a different effector, right? For the shivering, we think about muscles. For the sweating, we think about glands, especially our sweat glands. So they start to produce liquid on the surface of your skin. And then the evaporation of that liquid basically pulls heat away from your body, pulling you back down. So we have negative feedback loops in both directions. Questions about this process? Most important thing to carry forward for the rest of this course is really this idea of negative feedback. As we walk through the book and in the slides, you're often going to see these kind of like blocked out diagrams of processes. This happens, then this happens, then that happens. And they'll have little dotted arrows on them or red arrows. Sometimes they be labeled negative feedback, positive feedback. And that's an indication to you to tell you that they're tracing through some change and that there is a mechanism controlling it. So then you would go through and see all the opposite changes as we return to our set point for that. And I'll walk you through those as we start to see that type of figure, but negative feedback especially comes into play a lot throughout the course. So that sweat shiver, where we have different responses on opposite sides of the set point, we think about as antagonistic effectors. So basically we can move our regulated variable in either direction. Right? So we have two things kind of pulling or pushing as you want to think about it from both sides. And this is how we can partially maintain some of our consistency, is that we can move up, we can move down as necessary on this graph. We do have this in pretty much all of our systems where we're going to see regulated variables. So we have ways to raise blood glucose levels. We have ways to lower blood glucose levels. We have ways to raise blood pH. We have ways to lower blood pH because we're trying to stay within that normal. This is not directly from your book, but this is kind of an example of what those figures I was describing would look like. It's just from a different book. So we're thinking about glucose. Right, we see an action we might take is eating. Right? So when you eat food, say you eat a donut, right? Got lots of sugar and carbs in there. We are about to move into biomolecules. This would raise your blood glucose as you ingest, digest, and absorb the sugars from that meal. They get into your blood sugar goes up. We don't want our blood sugar to stay up way, way, way high. Right? We want to bring it back down to its set point, to its normal range. So we have organs that take care of this essentially. So in your pancreas, you have some glands. The pancreas is a gland. We'll talk at length about which parts of it do what. But we have parts of it that produce insulin. Okay? So we'd start to see an increase in insulin being put out by your pancreas and therefore ending up in your bloodstream. Once you have this insulin present, the whole reason we're tracing this loop, right, is we're tracing through a regulatory mechanism. So this insulin is going to help us bring that blood glucose back down. But insulin is part of our negative feedback loop. So what it actually does is it tells your cells to pull in a bunch of glucose. You have lots of glucose in your bloodstream swimming around in your cardiovascular system. Then you have insulin. It tells your cell, hey, grab some of that glucose. So as they start to pull glucose out of your bloodstream, what's left decreases, so your blood glucose drops. So this is all negative feedback. And then when we see this minus sign, this is kind of telling us how this gets turned off. Okay. So what would happen then is once we have our drop in blood glucose, right, we go around, we see, okay, we're going to stop producing insulin, means we're going to stop the cellular uptake of glucose. So that could return us to our set point. Or if we drop too far, we come over here and follow through what happens when we see a drop in blood glucose. So here you can see we've taken this same arrow but reversed it. So this is that turning off mechanism. All right, so we've reversed this arrow as well. But because systems are complex, we actually have a second system that comes into play to raise that blood sugar, which is a second hormone, glucagon. And it affects the liver and tells the liver to put some more glucose into the blood. Now, you don't have to memorize that specific example of all the pieces that work with glucose yet. We will talk about that in detail in one of our future units. What you want to be able to do at this point is to identify, if I give you a description of a negative feedback loop, I want you to be able to identify that that is a negative feedback loop, and be able to sort of identify, okay, what was our change, what was our stimulus, what regulated variable were we talking about, and what effect were we having, right? What was our effector at the end? What was our end result? That's our goal for right now for these scenarios. Quick note about positive feedback. Positive feedback does exist, but it's much less common in the body. We're usually much more concerned with keeping a situation stable. That's really the homeostasis piece, right? Trying to keep consistent internal conditions in the body. Okay? So in general, when we see a change, we want to shut that down. And that's the role of negative feedback. However, under certain conditions, we do want to accelerate a change. This is, for example, like ovulation work. So we'll talk about it as it comes into play in certain systems, or as our example here on the slide is, we have formation of a blood clot. All right. So in this case, okay, you got a cut on your hand, on your arm or something. Right. What starts to happen is you start to have that seal over. You have healing processes, which we'll also talk about this year. Okay? So you want to start to form a blood clot. If we were talking about negative feedback, as we start to see that blood clot form, like that's a change to the body, negative feedback would have us just dissolve that blood clot, right? But that's not what we want to happen in the case of healing over a wound. So what we have happen instead is when a blood vessel gets damaged and that clot starts to form, our regulatory mechanisms instead say, yes, great, keep making that blood clot. Right, so we'd have time here. Let's say this is going to be like the size of our blood clot on the Y. Right, so we have our little blood clot form here. Over time, it grows, and we want it to ramp way up and get bigger and bigger and bigger. Okay. So that would be our example of positive feedback. We see a change in one direction, and then we have mechanisms in the body that are amplifying that change and making it bigger and bigger and bigger. However, if we think about blood clots, you don't want a giant blood clot in your arm forever. Eventually that blood clot does break off, dissolve, and you go back to your normal body with your new healed blood vessel. So positive feedback is usually going to be followed eventually by some form of negative feedback. Another example we have is uterine contractions during childbirth. So we'll talk about them as they come up, but your best bet usually is going to be negative feedback. We're usually talking about, at least in this course, how we maintain our normal processes. And here's just another example of positive feedback. This is one from our book, but all this is telling you with this positive feedback is that we have a gland up by our brain, pituitary gland, secreting something called LH, luteinizing hormone. It's the same as your ovaries make estrogen. Because this is denoted positive feedback, if we were then to try and draw the following time step, we'd go through. We'd say, oh, okay, so estrogen is doing positive feedback. So we'd have more LH secretion in the next step, and we'd have even more estrogen. So that's another example of kind of how you do these type of things. And we will talk through them as we see them. Next thing we're going to think about as we think about regulation is basically like the level at which we're regulating something. So what we were talking about when we went through this figure with the man standing up is mostly thinking about extrinsic regulation. Okay, so we went all the way up to the medulla oblongata in the brain for our regulation. Okay. So if something far away regulates or is involved in that regulatory process, we think about it as extrinsic. Okay. When we think about intrinsic regulation, what we're talking about is that a cell within an organ senses a change and then immediately kind of changes back itself and signals to neighboring cells to make changes. So whether something is intrinsically regulated or extrinsically regulated for a given system is basically how far do we have to go to have this negative feedback take place? How far away are we getting these signals from? So when we talk about extrinsic regulation in our different units versus local or intrinsic regulation, extrinsic pathways are going to involve other organs than the one we're talking about. They're going to involve nerves signaling from one place in your body to another. Intrinsic regulation is going to be really right there. So we'll often be looking at the membrane of a cell and maybe the neighboring fluid, maybe the blood. And that's about it when we're thinking about intrinsic regulation. So we'll be highlighting as we go through when we're talking about something intrinsically versus extrinsically regulated. Okay. So we are going to move straight into our next packet, into our review of biomolecules and enzymes. You've seen these before in your previous coursework. So we're going to be going through our different classes of biomolecules. So our carbohydrates, our lipids, our proteins, and nucleic acids. We're going to be thinking about the structures and functions. And then we're going to zoom in and think specifically about enzymes, specifically about one of our types of proteins that is going to be really important in some of our chemical pathways throughout this course. When we're thinking about biomolecules or macromolecules for our purposes, those are going to be the same thing. This is a biology class. We're thinking about molecules that are synthesized by living things. They primarily contain carbon. So what I've drawn here is a reminder for you that carbon is going to form 4 bonds around it. So has space for 1 place to share electrons, 2 places to share electrons, 3, 4, which makes it particularly well suited to forming chains and rings. Okay, so we're going to have larger structures based around carbon. But we're also going to see other atoms present in our biomolecules. Okay, so we're going to be thinking oxygen, we're going to be thinking hydrogen, we're going to be thinking nitrogen for our purposes. With these components, we can make our 4 basic types of macromolecule or biomolecule. Our carbohydrates, so sugars or starches, they give us immediate energy. We make our lipids, so things like fats and oils that can be for long-term energy storage. Although I will say to you, lipids is a really actually diverse class. So we're going to consider anything a lipid that basically has non-polar interactions with other molecules. So we'll see that some hormones, for example, are lipids as well. We're going to have different groups within the lipids. Our proteins are going to be really the doers in our body. So they form our cytoskeleton, they form all sorts of receptors, they form our enzymes. They're prevalent in our bodies and they have a lot of different functions and activities. We're going to have our nucleic acids, so our DNA and our RNA that forms our genetic code and helps us build out those proteins. So we're going to start with the carbohydrates. So we want to be able to identify these. So when we look at a carbohydrate, we're going to see carbon, hydrogen, and oxygen. So if you have the chemical structure, you see an N in there. If you see a nitrogen, you don't have a carb. Okay. Just carbon, hydrogen, and oxygen. Okay. And we're going to divide them into types dependent on how big they are. So our monosaccharides are going to be essentially one ring. So we can have our carbons in a ring. So carbon, carbon, the carbon over here, carbon, carbon in each of the corners. You get the idea. With one oxygen here in that ring. Okay. And then we have our hydrogens and our hydroxyl or alcohol groups off of it. Okay. So this is what a monomer of a carbohydrate looks like. So if it's just one mono, we would call these our monosaccharides. Saccharide is for like sugar, so think saccharin, sucrose. Although sucrose is actually a saccharide, so maybe don't like sucrose. Okay. So we have our monosaccharides of just one ring, lactose, fructose, and almost always we're going to be talking about glucose. That's the real spoiler alert for carbohydrates in this class. That glucose is really our thing to know. Okay. So we want to be able to identify that. But we also can link together these monosaccharides into disaccharides, which have 2 monomers linked together, or polysaccharides that get big. So each of these in this glycogen molecule, a polysaccharide, is a separate ring of glucose. When we're looking at these larger structures, I like to think of carbohydrates as like these 2 rings holding hands with an oxygen in the middle. That's how they form. That's always how this looks to me. So that helps me identify whether we have this chemical structure with everything written out, or whether we have a big structure like this. Helps me kind of at a glance make my first guess that we're looking at a carbohydrate. So the way that carbohydrates are synthesized, so the way we form bigger carbohydrates, the way we form those polysaccharides, is by linking together our monomers, our monosaccharides, with covalent bonds. So this is the formation of that little holding hand structure with the oxygen in the middle. So this is actually a condensation reaction. So as our 2 monomers come together, we're grabbing this functional group, we're grabbing this functional group. We're going to take out water, we're going to release water. So we can go ahead and cross out. Here we have an H and an O. Water is H2O, so I'm also going to cross out this H. And that's how we end up with just that one oxygen in the middle with its covalent bonds to either side. When we form larger carbohydrates from monomers, we get water as a byproduct in this condensation reaction. Means we can also split them up Okay, so when we split them up, this is a hydrolysis reaction. So we need to put that water back in. We need those two hydrogen atoms and that one oxygen atom, put them in. And we can split those rings back up again. So you can imagine this happening as you're like stirring sugar into a glass of water, right? And it starts to dissolve. That's how I like to do it. Before we move forward, I'm going to give us some tips for the future. So part of why we're going through these different classes of macromolecules is so that we can start to understand, preferably kind of intuitively understand, how they're going to behave in water and how they're going to behave with respect to our cell membranes. So one important thing to notice as we're looking at these carbohydrates, okay, is that we're going to see all these hydroxyl groups, these OH groups. Okay, so they're going to have sort of little charges collecting on them. I think of oxygen as just kind of hogging electrons a little bit. It's electronegative, right? Creating slight accumulations of charges there, making these functional groups slightly polar, which is going to affect how they interact with the watery environment inside and outside our cells, our intracellular and extracellular environments. And as we move forward and eventually start to think about the composition of our cell membrane, the fact that these are here, the fact that we have this little bit of polarity is going to affect how they can and can't travel across that membrane. That's an important thing to start to pay attention to as we go through these different types of molecules. As we move forward to our lipids, the way we know we're looking at a lipid is it's going to be pretty much just hydrogen and carbon. When we're looking at the carbohydrates, we have like a 1 to 2 to 1 ratio of that hydrogen, carbon, and oxygen. Lipids may have an oxygen here and there somewhere, but it's almost entirely just these usually chains, right? So I will often just draw a lipid like this. This specifically would be a fatty acid. So I'm going to explain to you, if you haven't seen this before, what this kind of notation is. So every point would be a carbon here, as would every little valley. Okay. If I don't write anything else, you're going to just assume that there are hydrogens sticking off both sides. So this has implied hydrogen above and below that carbon. So that's how you get our 4 bonds for this carbon. So we have 1, the carbon on this side, we have 2, the carbon on that side, and then 3 and 4 are filled up by hydrogens. Okay. So carbon and hydrogen are similarly electronegative. They have similar poles on the electrons that they're sharing. So that is what creates this non-polar state for our lipids. We don't have really any accumulation of charges, which means that our lipids are going to be hydrophobic, and they're essentially going to repel water. So our examples for lipids are going to be formally triglycerides, ketones, phospholipids, eicosanoids, and steroids. But think about those triglycerides at first. Those are fats and oils, right? So think about oil, right? Like a layer of oil, if you drop some water into it, they don't mix, right? Oil and water very famously don't mix. That's because of this non-polar property of our lipids. Waxes will also be in this group. Okay. So that is really our unifying characteristic for our lipids. So our most stereotypical lipid that we will think about is our triglyceride. So this is why I began by warning you that you'll occasionally see an oxygen, okay? Because we do have a glycerol backbone, which is technically an alcohol. So we see an oxygen there And we do see an oxygen here at the beginning, right, as we bind into our fatty acid chain. But you'll notice that the rest of this molecule is just tons and tons of carbon and hydrogen and absolutely nothing else. That's how we would hopefully recognize this is a lipid. By that long chain and that just carbon, hydrogen everywhere. Our triglyceride is, or the glyceride part, that's referring to our glycerol backbone. That's this piece in the color orange. Tri for 3. That's telling us about the 3 fatty acids attached to it. So 1, 2, 3. Those are our fatty acid chains. So that is essentially what I was drawing with my lightning bolt squiggle thing. So those fatty acid chains are where we really see that carbon and hydrogen over and over. When we talk about fatty acids, we will differentiate our saturated fatty acids from our unsaturated fatty acids at times. So saturated, when we say something is saturated, I mean it's full. When we say we have a saturated fatty acid, what could it be full of? Hydrogen, okay? So what we're saying when we have a saturated fatty acid, we're talking about the fact that we have as much hydrogen in there as we can fit. So we have that carbon, okay, needs 4 bonds. So we have a carbon on one side, a carbon on the other side to keep forming that chain. And this is the one I was drawing from before, right? So we have a hydrogen here and a hydrogen there. We could not possibly fit any more hydrogen into this structure, so it's saturated. By contrast, an unsaturated fatty acid has kind of imaginary room for more hydrogen. So that's what we see below. So here, you can see a bit of a disruption in that squiggle form. So we have a carbon. We're going up to another carbon. But then instead of just coming down with a single bond to the following carbon, we have a double bond to the following carbon before our next single bond. So this means because carbon can only have 4 bonds around it, we have room for only 1 hydrogen on this carbon instead of 2. And same deal over here, only 1 hydrogen instead of 2. So this fatty acid doesn't include as much hydrogen as it possibly could, theoretically. So this is why we call it unsaturated. It's unsaturated because we could have a little more hydrogen to saturate it. When we're talking about nutrition specifically, we also often think about trans fats when we're talking about are they healthy, are they not healthy? Opposite of a trans fat would be a cis fat. All these words have to do with is where those hydrogens are positioned. Okay, so here we can see our trans fats have those hydrogens around the double bond on opposite side. So trans is going to be there, that opposite. The cis fatty acids, they're on the same side. So this is talking about the shape of the molecule and specifically the location of those hydrogens. We do have some more types of lipids. We have actually kind of a long list nested there in our lipids. So next one we might think about, we're kind of pulling after that nutrition piece, ketones. So ketones are also kind of a lipid structure, but really they're going to be what we see as we start to digest our fatty acids. So when we start to break down those triglycerides, some of what we're going to get is this structure called a ketone. So we'd have remaining groups at the R and the R. So you'd have variable kind of functional groups there. And you can see the double bond between the carbon and the oxygen where we had that attachment to the glycerol. And we'll talk about some byproducts as we get into metabolism. Think about how we break down triglycerides, how we break down glucose, how we break down proteins. Those ketones are related to our triglycerides. The other, I would say the most important of our lipids, I almost think of it in a separate category, but chemically we want to think of it as a lipid, our phospholipid. Okay, so our phospholipid is made up of a phosphate group plus lipid structure. Our phospholipid is going to have a polar head and it's going to have non-polar tails. So those are going to be fatty acids, essentially. And these different parts of the molecule are going to behave differently. So the reason I kind of think of them in my head as almost their own thing is that our lipids are non-polar, they repel water. So that's what's happening with our fatty acids down here, our fatty acid tails. But the head of our phospholipid, our phosphate group, is polar, so it's going to be attracted to water. Okay. We will pick up here next time, but this is what gives it an amphipathic property. So it interacts differently on each end, so it can interact basically in two ways with water. So the tails are going to be hydrophobic. The head, they're going to be hydrophobic. The important thing to understand when we start to talk about. Okay, we'll pick up here next time. Yeah.